Meiosis is the division of a cell that creates 4 haploid cells that are called gametes. These 4 cells are used in sexual reproduction. Meiosis consists of 2 stages, meiosis 1 and meiosis 2. Meiosis 1 is the crazy one and has processes such as crossing over and individual assortment occur. These processes make meiosis's daughter cells genetically different from not only the parent cell but also from the other 3 daughter cells made. This variation produces an advantage of meiosis. However, this process of meiosis takes much longer than mitosis, and that is the disadvantage. The second step of meiosis is meiosis 2, and this step is extremely similar to mitosis. Meiosis 1 begins with a phase called interphase. Interphase is where the cell will spend 90% of its time. Interphase consists of 3 stages, G1, S, and G2. G1 is where the cell will live normally. S is where the DNA will be replicated. The DNA right now in S phase is in chromatin, which is a loosely bound form of DNA that has some histones in it. G2 is where the cell will start to prep a little bit, only a little bit, for meiosis, and just organizing some things that weren't organized in S. The next phase is prophase 1, and during prophase 1, the chromatin begins to condense into chromosomes, and the nuclear envelope begins to deteriorate. Now we have homologous chromosomes that have 4 sister chromatids. You have 1 chromosome from your dad and 1 chromosome from your mom. The next phase is metaphase 1. This is where the homologous chromosomes line up at the metaphase plate. This is also where crossing over may occur, and also where the homologous chromosomes, 1 from your mom, 1 from your dad, go through individual lining up or individual assortment. The next phase is anaphase 1. Anaphase 1 is the phase at which the homologous chromosomes separate, however, the sister chromatids remain attached to each other. The next phase is telophase 1 in cytokinesis. During this phase, the chromosomes go to the opposite ends or poles of the cell, and the cleavage furrow begins to pinch in to create 2, not haploid yet, cells. The nuclear membrane begins to form just a little bit.